Okay, so we started using this new tool called NextBio. Has anybody heard of it? No? This was a bioinformatics company that was bought out by Illumina. And Illumina does pretty much 90% of all the sequencing on this planet. This software, what they wanted to do was, it's really easy to generate the data. The hard part comes in the interpretation of that. So what they did was they bought this bioinformatics company. They thought if they could form a company that would allow the researchers to get more out of their research, that more people would sequence. And they make more money on the sequencing than they do actually the interpretation. So it was kind of nice. What sets this apart from IPA, which I'll demonstrate later. Oh, wait a minute. It'll show up here. What they have done and what makes this special is that they have gone out into Geo and started basically curating all of these expression omic data sets. Not only just gene expression, but mutational, variations, SNPs, everything. At this point right now, it has over 20,000 curated data sets. What is nice is that I trust them to process the data correctly. They generate it, they can process it. So all I have to do, and what you'll see is, all you do is push buttons and you can generate your gene list. Not only that, but you are mining, in some cases, millions and millions of dollars worth of research for free. Well, you got to pay for the software too. But so here, and what's nice about this is not only can you use that information, so you could basically upload your gene list and compare it to everything they have downloaded in their database. You can also search for genes, and we'll actually do this at the very end. So at the very end, what I'll do is I'll take questions on, if anybody has any bioinformatic questions, or say you have a particular gene, we can kind of use this to see what it might say. So for this class, we were interested in breast cancer. So what I had the students do is say, okay, well, let's type in breast cancer. Okay, as I start typing it, all these phenotypes, these predetermined categories show up. The top here is just plain old breast cancer. So I click it. Takes a while. So what shows up is basically this kind of quick view summary page. What it's doing is it's based on its own information and all the curated data sets. It already has a group of genes that it knows is correlated to breast cancer. And I'll kind of show you that later here. It also brings up all the studies that have breast cancer as a phenotype. And according to this data set has, actually, let me go to that. 945 studies. So what I can do now, and obviously I can't go through all of those, but I can do my analysis on the fly. Well, actually, let me show you one more. Well, actually, I'll do that. So what I'm going to do is filter on this website. So I can go here to filter by. Can everybody see that? Is that okay? Okay, cool. I told them, well, let's stick with people. So you can just select all those studies that involve humans. You can go to data types, and you can see all the different types you can select from. There's RNA expression, SNP, histone methylation, you got mutational information, lots of things to choose from. We can select them all, but I just wanted to look at RNA expression. Now, as I mentioned, we were more interested in stage 3 breast cancer. So I can do that. I can actually look for those studies that have stage 3 somewhere in the description or in their gene list. So I put stage 3 and hit apply filter. Now we're down to 541 studies. This is how easy it is to make a list or to find genes that are associated with this particular, what we're looking for. You can see all these, basically the studies are here. So these are the public data sets that NextBio has gone and curated that we can now look at. So you can basically click this arrow here, and it'll give you a list of the comparisons within that study. You can see here stage 3 versus stage 1. Yep. So I select that. That gene list, which contains 7,226 genes, is now in my meta-analysis. And I'm going to show you exactly what that is. But I'm going to just generate a quick gene list to show you how easy it is. Again, what I'm looking for is stage 3 or grade 3, oh wait, versus grade 1 or grade 2. Stage 3, stage 1, yep. Go down here, select another one. As I'm doing this, this is basically contributing to my meta-analysis. And again, as you can see, it is incredibly easy. Get some more, hold on a sec. Stage 3. Nope. And because I put that keyword in there, T3, I think T is kind of this European version of stage. Oh, how many do I got there? So just me clicking, I now have 6 gene lists to look at that are associated with, basically are genes that are different between stage 3 versus either stage 2 or stage 1. Does everybody understand what I just did, or any questions on that?
Everything that they have in there is from Geo.
Or some other public data set.
Okay.
And they also have incorporated some private labs as well. So a lot of they partner with biotech companies that will have their own data sets, and they will actually, they have an agreement where they can use their data.
So it's more than just the publicly available.
Yes. So for the first, the first time I taught this class, we did lung cancer, which is, it's funny, when you ask high school kids, think of a disease, they always say like cancer. Which might be suggestive that I think cancer is getting worse. But, we'd have to go in and actually go into Geo, download the data sets, and analyze them ourselves. With this, I just did that with a push of a button. Okay? So now I have all of these in my meta-analysis. Let me click on the meta-analysis. Here are my lists. Actually, I didn't get any stage 2 in there. So this will be stage 3 versus stage 1. Again, this is showing me I can remove any list that I want. I can add them at any time. It's very, very flexible. Now when I click on this next tab, this is the gene results. This is going to give me, ordered by how many times this gene is differentially expressed in these different gene lists. It'll order it by that. And it'll also tell me what direction these genes are going. So in this case, this cool inducible RNA binding protein seems to be down in stage 3 versus stage 1. It is down significantly in 5 out of the 6 studies that I just put in. And you can see there's a lot of agreement here. Well, some of them. This is kind of odd here. But again, you can see you're almost using the crowd to kind of finalize your gene list. As you're going to see so we did this, and we used 24 gene lists. When we finally, and I'll go into, basically how we made that gene list, but it's very easy to export this gene list as a Excel table. You can also, just on this page alone, you can get biogroup results. So this is going to give you some kind of preliminary bioinformatics information on this. Come on. So as you can see, there's a lot of genes involved in the cell cycle. Yeah?
Is the specificity, does that mean if it showed up in 6 out of 6 of the studies that you're analyzing?
Yes.
Okay.
Totally. Yep. That means it was significant that you wouldn't expect this many genes due to random chance associated with cell cycle in all of these studies. Not only that, but the direction of these genes, what this is telling me is that these genes that are associated with cell cycle tend to be on average more active, upregulated. And again, you can go down here, that's just giving you interface. You can see all the cell division stuff in here. You can go to bioset results. Oh wait, I don't know if I'll give it that. Oh, these are different gene lists, I think. All right. Let me show you what we did. So we did this meta-analysis for breast cancer using that filter. We got 24 gene lists, and I downloaded that gene list, and this is what it looked like.